Hello everyone, welcome back to my CellRank video tutorials. In first and the second video, I showed you how to set up kernels and create a RNA velocity and pseudo-time transition matrix for cell rank analysis. So in this video, I'm going to show you how to use the estimators and computing the initial and terminal cell states. In order to repeat the online tutorial, I'm going to first run the data process, then we can identify the initial and the terminal cell state. So first we can load the packages. Then we load the unprocessed data. So now we can quickly process the data according to the online tutorial. So after pre-processing the data, we can run scVeno to perform RNA velocity analysis. So now the data is ready to perform cell rank analysis. First, we can set up the velocity kernel and the computer transition matrix. So we will use the data for next video tutorial. So we can save the data. The online tutorial process the pancreas. First, we can write the transition matrix into the data, then we can save the data. So now we are ready to initialize the estimator. The estimator uses the GPCCA method. It stands for the generalized parent cluster analysis. We can initialize the estimator and also have a look at the estimator. We can run. You can see we have the GPCCA for velocity kernel. In this dataset, we have 2,531 cells, but at the moment, we don't have the initial cell states and the terminal cell states. So next, we can run a cosine analysis to identify all the initial and terminal cell states in this dataset. First, we can ignore the warnings. In this dataset, we have 7 cell clusters. If we consider each cell cluster as a cell state, then we can set the end state as 7 to run the cosine analysis. Let's set the end state as 7 and have a look at all the possible cell states in this dataset. Okay, you can see it generated a figure. We have 2 NGN3 no expression progenitor cell clusters and 2 NGN3 high expression progenitor cell clusters. Then we have the epsilon, alpha, and beta cell clusters. So when we set the end state as 7, we identify the 7 cell clusters. But you can see here in this dataset, we have a terminal state cell cluster delta cells. If we set end state as 7, we didn't identify the delta cells. So we can increase the end state until we can identify the delta cell cluster. So we can increase from 7. I tried. If we set end state as 9, then we can identify the delta cell cluster. Let's run the code for end state as 9. You can see now, if we set end state as 9, we have 9 cell clusters now, and now we have the delta cell cluster. Because we have the knowledge about the pancreas development, we know the beta, alpha, delta, and epsilon cells are in the terminal stage, but we set the end state as 9, so now we have subcell clusters for the NGN3 low and high expression progenitors. So we can have a look at the estimator now. You can see we identified all the possible cell state, but we haven't performed the initial state and the terminal state analysis. So for the coarse-grained analysis, it will generate a coarse-grained transition matrix data. So this coarse-grained transition matrix data will be used to classify the initial cell state and the terminal cell state. We can have a look at the coarse-grained transition matrix. Let's make a plot. You can see this is the plot for the coarse-grained transition matrix data. You can see we can see high value for epsilon, beta, and alpha cells. So they are in terminal state. And the NGN3 low expression progenitors subpopulation 1 could be the initial state to give rise to epsilon, beta, and alpha cells. Because we set the n state as 9, so we have the delta cells here. So possibly the NGN3 low expression progenitors too give rise to the delta cells. So we have the coarse-grained transition matrix. Next, we can predict the initial cell state. And also, we make a plot to show the initial cell state. We can run the code to predict the initial cell state. You can see we generated the figure. You can see NGN3 low expression progenitors 1 has been identified as the initial cell state. If we have a look at the estimator now, you can see now we have the initial state as NGN3 low expression progenitors subpopulation 1, but we don't have the terminal cell state at the moment. So we can confirm the initial state identification by the expression of marked genes in the ductal cells. If you watched my previous video tutorial, I mentioned that the progenitor cells also generate the ductal cells. The ductal cell cluster can be seen in the SCVino online tutorial, but this cell cluster was removed in the CellRank tutorial. So cells in the initial cell state should express the ductal cell mark genes SOX9, ANXA2, and BICC1. We can run the embedding to see the expression of 3 ductal mark genes. Let's run. You can see all 3 genes, they have high expression in the NGN3 low expression progenitors. Because we set the n state as 9, we identified 2 NGN3 expression progenitor cell clusters. You can see subpopulation 1 has a high expression for all 3 genes, then subpopulation 2 express the lower level of 3 genes. So, which means our analysis is correct for the initial cell state, because the initial cell state give rise to endocrine cell clusters and also the ductal cell clusters. So next we can identify the terminal cell states. I showed you in the coarse-grained transition matrix already. The data only show high value for the epsilon, beta, and alpha cells. If we run the code, let the CellRank package to automatically detect the cell state, we only identify 3 terminal cell states. Let's run. You can see on the figure, we only identified the epsilon, alpha, and beta cells as the terminal state cells. We don't have the delta cells. So we need to set the terminal state as alpha, beta, epsilon, and delta cells. Then we run the terminal state again. You can see now we have the terminal state for alpha, beta, epsilon, and delta cells. If we have a look at the estimator, now you can see we have the initial state as NGN3, no expression progenitor subpopulation 1. Then the terminal state has alpha, beta, delta, and epsilon cell clusters. We can confirm that the terminal state identification by the expression of well-known mature cell type specific mark genes for endocrine cells in the pancreas. Let's run the plot first. You can see the glucagon is the mark gene for alpha cells, insulin is the mark gene for beta cells, somatostatin is the mark gene for delta cells, and the ghrelin is the mark gene for epsilon cells. From the mark gene expression, we know we identify the correct terminal state cell clusters. If you look at the UMAP for the beta cells, it identified the beta cells in this area as the terminal state, but for beta cells, you can see here, we have some cells express high level of insulin and some cells express low level insulin. Only high level insulin expressing cells have been identified as the terminal state for beta cells. So we can further confirm the terminal state for beta cells. To do this, we can compare the insulin expression level in the terminal beta cells compared with the other non-terminal beta cells. So we can subset the beta cell population from the A data, then label terminal beta cells as terminal and other beta cells as not terminal. Then we can make a volume plot to compare the insulin expression in both cell clusters. Let's run. You can see we generated the volume plot, and the insulin expression in the terminal cell clusters is much higher than the non-terminal beta cells. So again, we use the volume plot to confirm the terminal state for the beta cells. You can do this for other cell types. Okay, that's today's analysis. If you look at the online tutorial divided the analysis to a basic and advanced analysis. So I combined both methods in my video tutorial. Hope this is much easier for you to understand it, and then you can perform your own data analysis. So I'm going to stop here for today's demonstration. In my next video tutorial, we can estimate the fit probabilities and driver genes. I hope to see you in my next video tutorial.